Hello, everyone. My name is Jacquelyn Lee, and today my fellow scientists, Lucy Gworek, Jenna, Pham, and I will be focusing on our design drug containing functional green fluorescent protein as a cure for skin cancer. So there are many different types of skin cancer, and most skin cancers are characterized as a fast spread of uncontrollable cell growth. Here we are focusing on basal cell carcinoma, also known as BCC, because it is the most common type of skin cancer with over 3.6 million cases per year. And we are emphasizing on mutations of the PITCH1 gene. We are determined to find a cure for BCC as a step attempting to advance one field of cancer is a step made to advance all fields of cancer. As most of you already know, cancer can be one of the most vigorous diseases an individual can endure in their lifetime. Some cancers are treatable, whereas some are not. Cancer might start in one region of the body, but it is very easy for it to spread to other areas and damage those tissues as well. For this reason, our goal is to not only find a cure for BCC, but to advance the field of skin cancer in general. We designed the LJJ drug using a pre-existing topical treatment with the addition of green fluorescent protein isolated from the rare Amazonian plant Green Glow Funkadelicus, also known as G-Funk. And for this reason, and in doing so, we will examine the impact our drug will have on BCC tumor death. A little bit more about basal cell carcinoma. So BCC is one of the most common types of skin cancer, like I just said, and this cancer directly causes irregular basal cell growth. Basal cells are skin cells that make up the outermost layer of the epidermis, and damaging these cells will harm deeper tissues of the epidermis and possibly damage other parts of the body. Our skin acts like a barrier to all the toxic particles floating around in the environment. Therefore, we don't want to damage the epidermis. BCC can cause growths of tumors directly on the surface of the skin, most commonly on the face or around the neck area. And tumor examples of tumors include redness and soreness around the infected areas, open sores, and swollen or elevated sores. It can be as painful as it looks on the skin, and most treatments will enhance the pain or cause more irritation before progress is made. The cause of BCC is most likely due to the overexposure of UV radiation. With all the different types of UV radiation, UVA and UVB radiation is the reason for abnormal cell proliferation. UVB radiation reaches the epidermis while UVA reaches the dermis or deeper tissues of skin. While UV protection is very accessible, the exposure of UV radiation is inevitable, and the effects have a lot to do with skin cancer, or skin color, sorry. People of color are less likely to be affected by UV radiation because they have a higher level of epidermal melanin that provides photoprotection, whereas people with a lighter complexion will be less likely affected because they have lower levels of melanin. Any type of UV radiation will lead to an increasing rate of mutations, and most common mutation leading to BCC is mutations of the PITCH1 gene. Compared to other cancers, BCC is very manageable because the rate of cell proliferation isn't as rapid. However, advancements in treatments for BCC still hold importance because high degrees of BCC can lead to damage of bones. It is most important to treat the cancer that is currently in patients and not let it spread and become different forms of cancer. Therefore, finding a cure to BCC will be extremely beneficial. The green fluorescent gene is something that has been discovered for many years. However, we did not know this gene could possibly contribute to the research of BCC. Originally isolated from jellies, Aquaria victoria, this gene naturally expresses a fluorescent that acts as a defense mechanism against predators. However, as of recently, this gene has also been found in the Amazonian plant G. funk and is used by natives of Brazil as a remedy for skin wounds. Upon this discovery, we knew that the gene or the green fluorescent gene has already been sequenced, and seeing that it has healing abilities and it provides an anesthetic to the primary wound, we wanted to incorporate this into existing treatments for BCC in hopes that it would be more effective than the pre-existing treatment. And we will use the already sequenced green fluorescent gene to produce functional green fluorescent protein.
So in order to properly research the effectiveness of the green fluorescent protein, we would need a lot of it so that we could do lots of different studies. To do this, we used a common molecular cloning approach. First, we extracted the DNA of the G. funk plant and restricted our gene of interest, the green fluorescent protein. We used E. coli cells to grow our piglo recombinant, and once we were sure we had a working GFP, we used PCR to mass produce piglo. We could now take that piglo, re-restrict the GFP, and mass produce that. So we were successful in mass producing the GFP gene for our use. So there is this drug, fluorouracil. It is a pre-existing topical skin cancer drug. It is effective. However, anything that is somewhat effective can always be improved. So fluorouracil does come with side effects such as burning, itching, rashes, oozing, sensitivity to sunlight, soreness, redness. It also has a 12-ish week time period, which is its rate of apoptosis. So fluorouracil is an anti-metabolite drug, and it works by killing abnormal cells, in this case, tumor cells. So what we wanted to do, or the objective of this study, is to investigate the synergistic effects of fluorouracil with GFP on apoptosis of BCC cells, as well as seeing if we can soothe any of those fluorouracil side effects. We hypothesized that GFP will improve the side effects of fluorouracil, as well as speeding up rate of apoptosis. We believe this to be feasible because we understand that GFP is already known to have anesthetic properties as well as skin healing properties. So with this knowledge, we could see that having GFP mixed in with the fluorouracil treatment will soothe things like skin rashes, pain, soreness, redness. We believe that it is possible that it will speed up apoptosis because it acts as a healing agent. So as we're killing cells, we're also healing the skin around it. So that's what we hypothesized. This matters because although we have this topical treatment that works for BCC, in some cases, it is the main option. In cases where it's on, like, say, the back, surgical removal of the tumor is possible. But in cases where BCC is on the face, fluorouracil is really our only option for treatment. And we'd really like to see this improved. We'd love to have it be a sure thing that we can get rid of these tumors. BCC on the face can cause permanent mutations and disfigurement of the face. And we're hoping that if we can speed up the rate of apoptosis, killing these tumors faster would lessen the likelihood that someone would be permanently disfigured from BCC.
So for our experiment, we chose mice as our model organism. And the reason for this is because they are very easy to breed. They are easy to keep up with, and they have a very high similarity of genome to humans. So for the mice, we purchased 2 wild type pitch 1 mice and 2 homozygous knockout pitch 1 mice to breed. We bred enough mice to have a total of 120 mice as a population. And it is important to note that we will continue to breed the mice throughout the entire experiment, just because there could be cases where mice will be sacrificed or euthanized. Therefore, we want a constant availability of mice. And so with that, there will be 8 tested groups of mice and 15 mice in each group. 4 of these 8 groups will include the wild type, and the other half will include the mutant pitch 1 mice. We will test the effects of no active ingredients, which will only be a topical cream made of shea butter. And mice will also receive only fluorouracil, mice that will only receive only GFP, and mice that will only receive our drug, the LJJ drug, containing 0.05% active fluorouracil and 1% active GFP. All of the treatments that are given to the mice will be in the form of a topical cream.
So as the groups Jacquelyn mentioned before, there are 8 with 15 in each of them. The first group will be the wild type with fluorouracil. The second group will be wild type that is only treated with GFP. And the reason we're doing this is because we want to test the effect that it has on the mice so that we can proceed to group 3, where we have the wild type treated with our LJJ drug. And then finally, group 4, for this wild type group, they will receive a placebo treatment of just shea butter. So in group 5, it will be the heterozygous mutated mice that is treated with just fluorouracil. Group 6, treated with just GFP. Group 7, treated with our LJJ drug. And group 8, just the placebo treatment. Next. We can now begin our experimental protocol where there will be mice chosen that will be monitored until a tumor size of about 70 to 100 millimeters cubed has formed. All mice that aren't chosen will be euthanized immediately. We want the tumors to form on their dorsal side, and pictures will be taken at the beginning and at the very end to compare. Treatment begins twice daily for 12 weeks, once at 7 a.m. and once at 7 p.m. It's for 12 weeks because this is typically the longest extent of the fluorouracil treatment. Each group of mice will have proper husbandry of having their own cage, food, water, and consistent temperatures. Data collection will begin at the 3-week mark, and then at the end of each week. Before getting treated on Monday mornings, each mouse tumor will be biopsied for flow cytometry to measure the apoptosis of the tumor cells. We'll also be monitoring the appearance of the tumors by observing for skin redness, rashes, and oozing and pussing. Like I said before, we'll also be taking pictures each week to document the tumor.
So for collecting data for the apoptosis of cells, as Jenna was mentioning, each week the tumors of the mice will be biopsied and undergo flow cytometry. So each week we'll take the average count of apoptotic cells for each group, and we'll graph this against time lapsed. In doing this, we can compare and contrast apoptosis rates for each group to see if one group had a higher rate of apoptosis, hopefully our GFP fluorouracil group. We can also look at whether our group treated with only GFP experienced apoptosis, because we have groups that, like our placebo group, is not gonna experience the rate of apoptosis that a treated group will. So hopefully our GFP alone could do something that would be a really great outcome. For data collection for our side effects, for characteristics. So as Jenna was saying, daily someone will record the characteristics of each mouse's tumor. We're looking for things in like rashes, redness, oozing, and we'll be able to compare our GFP or LLJ drugs, so our GFP fluorouracil group, to just fluorouracil to see if GFP helped take down any of those side effects. We're also looking for any other things, any other notable things. So the GFP only group, we're hoping could have some positive effects, shrinkage of tumor, etc. So we'll be looking at all of those things and collecting data on that daily.
Okay, so we have already began FDA approval steps for human use. The preclinicals were the addition of GFP into the already FDA-approved fluorouracil ointment known as Fluoroplex cream that was applied onto the mice. Once the LJJ drug is produced, we can start clinical trials over a 12-week period. And once we see that the mice have been successfully treated and the treatment was safe, we can proceed with FDA filing and clinical applications where we want to produce a large quantity of the LJJ drug. We want to get FDA approval in human clinical trials. Once the human clinical trials are proven to be safe and effective, we can get final approvals to start distribution and create enticing marketing ideas for the public. So what do we hope to achieve from all of this? We want a higher efficacy rate with the addition of GFP to fluorouracil because fluorouracil itself is currently 93% effective, but we want it to be closer to 99 and 100%. We also want to decrease the harsh side effects that come with fluorouracil. This treatment can heavily advance the skin cancer community as more treatments based off the LJJ drug can be developed to target other types of skin cancer and maybe even a broader range of cancers besides the skin. We want to ensure people that this new form of treatment will have very positive results. However, we do run into some problems. Fluoroplex with the addition of GFP only targets early basal cell carcinoma, but if the cancer has advanced, it won't do much. So a common treatment is surgical removal, but like Lucy previously mentioned, if it's on the face, that isn't possible. So a topical treatment called patidegib is being developed with a mechanism called hedgehog pathway inhibitors, which essentially targets the advanced basal cell carcinoma. We believe that the addition of GFP to this drug could potentially solve this problem of advanced basal cell carcinoma.
So before we close out this presentation, I just want to really reiterate some important parts. Cancer, not just skin cancer, but all cancer is something that impacts all of us, people as a whole, the entire world. So any type of cancer research is important. Although this specific research is done on a very niche type of skin cancer, BCC, I just want to say that if we can find a treatment for this, we could broaden that and use it in other areas. Medical research in one area often overflows into other areas of the medical world. So where else could we use this? Yes, this is a topical treatment. Could we use this in future inside the body in some way? Learning about this now in any small amount could change how we treat cancer forever for the future. I think it's really important to note that it's got anesthetic properties, so pain relieving properties, which there's a lot of painful treatments for skin cancer. So this is definitely something that we want to look at more at in the community of science.
I would like to say thank you so much for listening to our presentation. And I really hope that this has made an impact on what you think skin cancer really means. And I hope that you got the bigger picture out of our presentation for how this will affect the community as a whole.
So we really hope that from our presentation, we get the funding that is necessary to help skin cancer and potentially all cancers in the future. Thank you.
Yeah, thank you so much for your time. We really hope that you choose to fund our project.